Okay, so yeah, I'm going to talk mainly about myosin, and warning, there's no machine learning or language models in this talk, which there have been a lot of so far today. And I should say, a lot of this work has been done by, that's odd because I don't see the same slides as on here. Okay, can't go back to the title slide. So you can't see the picture of Jake McGreek, who was my PhD student. He's sitting there. He did the computational work, and Chloe Johnson, PhD student, who did the wet lab work in this project. But you'll get to see their pretty faces at the end in the acknowledgement slide. So beta myosin. The talk is about this protein here. And it's about how during evolution its function has been fine-tuned to species size. So as species have evolved, they've got larger, and that's meant it's not changing. That's better. Okay, perfect. Right. So yeah, as species have evolved, they've got larger. Beta myosin is expressed in the heart, and as species have got larger, heart rate has reduced. So a mouse has a heart rate of around 300, maybe 400 beats per minute. An elephant has a heart rate around 30 beats per minute. Now, that's really interesting because this myosin, which I'm talking about myosin because that's the motor that's expressed in heart and muscle, and that's what's determining the maximum rate of contraction of a muscle. And it goes through this cycle. Now, it's all a bit complicated. I don't understand the cycle because I'm not a myosin researcher. But what I want us to focus on is that this is a highly regulated process. So it's highly regulated. All of those steps, they have to occur in the right way for muscle and heart contraction to occur. So the second thing from this slide is that this bit on the left here, the ADP release is the rate limiting step. So that's ADP release from the myosin is the rate limiting step in this whole cycle. And so that's what's responsible for determining the maximum rate of contraction of a muscle. And there's lots of experimental data that's showing that's the case. So our question was, well, in this highly regulated process, how has the myosin evolved so that we end up with a mouse that can have a heart rate of 300 and an elephant a heart rate of 30? So we started out with a set of myosin isoforms. So we've taken 13 different isoforms here. Some of them are expressed in muscles, some of them are non-muscle. And we then looked in each of those isoforms, and we used mouse as a reference because it was one of our smallest species. And we've compared each of the sequences to that mouse sequence and calculated the sequence identity, which you can see on the y-axis here. And then on the x, we've plotted the mass of that other species. So on the left, you can see the results for non-muscle A. So that's kind of a control. It's not a muscle myosin. We don't expect to see any correlation here. And then on the right, you see the beta myosin. So as the species get larger, there's more deviation from the mouse sequence. So over time, those sequences have been obtaining more changes. So we propose that some of these changes are probably going to be responsible for altering the ADP release rate, which enables the rate of heart contraction and muscle contraction to change. So summarizing over all 13 in this bar chart, and we saw that for 4 of the sarcomeric, so that's for 4 of the muscle myosins, there's quite a strong negative correlation with species, with size, and fairly high correlation coefficients. Then what we wanted to do was, well, what are those specific residues that are responsible for changing the release rate of ADP? So we took each of the position in the alignment where there are different amino acids, and we applied a logistical regression model. We took the two most frequent amino acids, most of the positions only have two, and we scored them as a 0 and a 1. And so you can see here two of these examples where there's a nice trend. So on this one on the left, there's an S predominantly in the smaller species, there's an M predominantly in the larger species. There's triangles and squares because we've got two main different clades of species that we considered. So clearly this is an example where this position, so position 326 on the left, there's clearly a change from an S to an M as the species get larger. But obviously for not all positions do we see that correlation. So in these top two here, there's no real relationship with mass. We can see both amino acids present across the whole range of masses. So if we look at the phylogenetic tree, I think what's interesting here is in our two clades, so if you look at the first two positions, these are the ones where there's no correlation and the amino acid change differences relate with parts of the tree. But then if we look at the second two, we can see that the differences in amino acids are spread in both clades. So we've got some form of parallel or convergent evolution occurring. So the same changes at the same positions are occurring in the different clades. So we performed some statistical tests to try and find out, well, which of these positions we really see some significant changes. We did that here, and we end up with a fairly small set of positions. Now, some of them are in positions that we know are hypervariable, such as these loops that are labeled, and we've ignored those. But we kind of end up with these three nice groups here, which we're going to look at in some more detail. But then we also wanted to think about, well, there may be some positions that are just associated with the clade, and we don't want to be identifying those. So we've done a test to see if the changes that we see relate to the clade. And so that's, sorry, that should be on the X. Anyway, this box here is where we have a significant association with the body mass, but not with the clade. And we've ended up with a fairly small set of around 12 positions that we're interested in. So what does that look like? Well, here I've selected a few of the species that cover a fair amount of that range. And it's been easily color-coded so that we see the amino acid that's present mainly in the smaller species is yellow, and the larger species it's blue, and there's this trend. And obviously at some positions there's a third or even a fourth amino acid present sometimes. Now, if we expand that to look at all the species that we considered, and counterintuitively, I put the smaller species here and the larger species there, and we can see this trend. We start with predominantly yellow, and we increasingly get more and more blue residues, and towards the end, it's all predominantly blue. But it's a trend. There's no clear rules. We even see some of these yellow-colored residues in some of the larger species, and we see some of these blue ones in the small ones. So we were quite confident that these positions are probably altering the release rate of ADP and therefore the rate of muscle contraction. Where do they occur in the protein? So the nucleotide binding pocket might be the obvious place to think that actually this is a highly regulated system and binding in of ATP is really important. We can't really mess with that. So the changes occur away from there, not necessarily too far away, but some of them are in positions where they may affect binding of actin, because binding of actin is what encourages release of ADP. And that's something that we're currently looking at. But what we wanted to do after that was, well, let's explore this a bit more. We've got these residues. We think that they have this role in fine-tuning the function so that the ADP release is altered. So we went to the wet lab and we made a chimeric beta-myosin. So we took the human protein and we inserted the residues at these positions, so those are the ones in red, from the rat. So we've got a human rat chimeric myosin. Now I'm told that was an awful lot of work. It's really hard to express myosins. They don't fold well in vitro. So yeah, anyway, what do we do then? So the first thing we did was perform an in vitro mobility assay. So we have a cover slip and we immobilize the myosin on that cover slip, and then we have fluorescently labeled actin. And when you add ATP, the actin will move on the cover slip, and you can view that. And now if this video works, which it did earlier, we can see on the left you have the wild type, and on the right you have the chimera. Now, the chimera, if it's more like the rat, it should be moving faster than the human, which is what I think we see here, which is a nice first result. So then we do some more biochemical characterization. So the top left graphs here are histograms representing the speed of those actin filaments on that cover slip. And we can see that the wild type, the blue, is much slower than the chimera. So that's the first thing that we're happy with. And then we've performed a range of other biochemical, biophysical experiments. First looking at ADP affinity, and the thing here again is that we have the human wild type in blue, and then the other two, the rat and the chimera, are more or less the same, so they have nearly identical ADP affinity. And then if we look at ADP release, which is where we're really interested in, we see that the chimera behaves in more or less exactly the same way as the rat sequence. So we conclude from this that these positions, these 9 positions that we've changed, have a role in altering the ADP release. And so over time in evolution, as species have got larger, they have resulted in that heart rate being able to be reduced. So, kind of just gave the summary, but anyway, it's really well established. We've seen that in mammals, in muscles of larger mammals, they contract more slowly. We've applied a bioinformatics approach to identify positions that are associated with this increase in species body mass. And that's also enabled us to identify these residues that are involved in fine tuning. And then we've carried that on and validated it with some wet lab experiments. So acknowledgements, I clearly didn't do all of this on my own. This is a really long time collaboration with Mike Jeeves, who is the myosin expert in this work. And as I said at the beginning, Jake McGreeg did most of the bioinformatics work. Chloe did a lot of the wet lab work, or most of it, I should say. Leslie Linewand at the University of Colorado Boulder, she was who we made the chimera with, and we used her lab to express the myosin. And then, yeah, thank you very much.
Very interesting thoughts or questions? Yes.
I really enjoyed the talk. I was curious, in the human population, are there no genetic variants that in myosin that might relate to this result?
So, yes, myosin has more than 300 positions that are associated with cardiomyopathies. So if you have a mutation in the motor domain of the myosin, which is the region we've been looking at, then it's very likely that you get a disease because your heart doesn't beat properly. Interestingly, we don't see really any overlap with the positions that we've identified here and those that are associated with disease. In fact, when we look in NOMAD, we see, I think, no variation at really any of these positions at all. So we have an example where the protein is very highly conserved within the human population, but it varies significantly between species.
Okay, maybe we can look at Neanderthal and genetic variants next.
That sounds exciting.
Other questions?
So these mutations away from the active site, is that allosteric effect, or is that the interaction with other proteins? You think that is modulated? And then if it's interaction, would you expect co-evolutionary changes on these interactors?
So the answer is we don't know, Jens. So that's what we want to do next, is find out what are these residues doing, what is the role. So there are quite a lot of them in positions that are not too far away from actin binding. So I think the actin binding site is down here somewhere. Yeah, these two sort of brownish colored regions. And so some of these are sort of on this helix there that's connected, and this region here. So that's very much our hypothesis, that there's some sort of allosteric effect, could be relating to actin binding, but we haven't done any of that work yet.